We saw earlier in this chapter that amino acids are joined together to form peptides. And remember that a peptide bond is nothing more than an amide bond, but it's between amino acids. And as an example, glutathione is a tripeptide, which is assembled from glutamic acid, cysteine, and glycine. And you see it here, so we have our glutamic acid, there's our first peptide bond, then we have our cysteine, second peptide bond, and then we have our glycine right here. Now, glutathione is found in all mammals, and it serves an important role as a scavenger of harmful radicals and electrophiles. Glutathione exhibits two peptide bonds, like I have highlighted here in yellow, and it's comprised of these three amino acids. So we have our glutamic acid, our cysteine, and our glycine. And when amino acids join together to form a peptide, the order in which they are connected is important. For example, consider a simple dipeptide that you could make by joining alanine and glycine. We could make the peptide bond formed between the carboxyl group of alanine and the amino group of glycine. So we see that here, carboxyl group of alanine, amino group of glycine, and there's our peptide bond. Or we could make a dipeptide using the carboxyl group of glycine and the amino group of alanine, and there is our peptide bond. But if you look carefully at these two compounds, you'll see they are not the same compound, but they are constitutional isomers. They have the same molecular formula, but they're connected in a different way. Now, peptide chains always have an amino group on one end, and we call that the N-terminus. So that's going to be on the left-hand side, and that's just a convention that we always put the N-terminus on the left-hand side. And then on the other end, we have our carboxyl group, and that's where we have our C-terminus, and that is on the right-hand side. And again, that's just a convention. That's the way we always write them. So in order to remember the way that you write out a sequence of amino acids, what I tell my students to remember is, you notice it goes nitrogen, carbon, and then a carbonyl, right? And then it goes nitrogen, and then a carbon, and then a carbonyl. And I'm not going to keep going from there, but you can see it always goes N, right? Nitrogen, carbon, and then a carbonyl, and then it just keeps repeating like that. Now, furthermore, besides actually writing out the structure of each amino acid, you can also use the three-letter abbreviations or the one-letter abbreviations, but it's important to remember that the N-terminus is always on the left-hand side. So you see this peptide here, which is comprised of 1, 2, 3, 4, 5, 6, 7, 8, 9, 10. So this would be a decapeptide. But it's important to remember that your N-terminus, right, your amino group, I can write that a little bit better, is coming off of your alanine, and your C-terminus, your carboxyl group, your free carboxyl group, is coming off of the leucine. In order to understand the three-dimensional geometry of peptides, we must first take a look at the geometry of peptide bonds. Recall that peptide bonds are amide bonds, and that amides have a significant resonance structure that endows the carbon-nitrogen bond with some double bond character. So if we take a look at all three of these resonance contributors for our amide, you notice that in the first two, the carbon-nitrogen bond here that I have highlighted in yellow is shown as a single bond, but in the last resonance contributor, which is a significant contributor, you can see we have a double bond here. And so that carbon-nitrogen bond does have some double bond character. And as a result, the nitrogen atom is sp2 hybridized, and it's planar. Now, peptide bonds are simply amides, like I keep saying, and therefore they exhibit double bond character just like regular amides, and as such, a peptide bond experiences restricted rotation, giving rise to two possible conformations called S-trans and S-cis. And you can see them down here. And the S, if you're wondering, what does this S stand for? S-cis and S-trans, because this isn't cis and trans like the traditional way that we know it, either in a ring or using an alkene. Well, the S actually stands for sigma, okay? So the S stands for sigma, right? Because we know this bond has more sigma bond character than it does, double bond character. And so there is a rotation around that bond. But when the two biggest groups are on opposite sides, we call that S-trans. And when the two larger groups are on the same size, we call that S-cis. Now, the S-trans conformation is usually more stable because it lacks the steric interaction that you see in the S-cis. Here you can see there would be a huge steric interaction here. Now in a polypeptide, each peptide bond will show a preference for adopting an S-trans conformation. Nevertheless, polypeptides are not entirely planar because they still possess a sigma bond that experiences free rotation. Only the peptide bonds experience restricted rotation. So if we take a look at the peptide bonds, which are highlighted here in green in these green ovals, those are the ones that have double bond character, but all of the other sigma bonds in the backbone, okay, all these bonds that I'm highlighting in yellow, these are just sigma bonds, and all of those bonds can freely rotate. And so we can have a variety of peptide conformations that are possible. Now, way back at the beginning of this course, we talked about thiols a little bit. And you might remember that thiols can be joined together via an oxidation process to form a disulfide. So if we have, let's say, ethane thiol, right, plus another molecule of ethane thiol like this, okay, and if you join them together to form a disulfide like this, so keep in mind this is called a disulfide, okay, this is actually an oxidation process, right, because the sulfurs are losing hydrogen atoms. So again, joining the two thiols to make it disulfide is an oxidation process. And of all the 20 amino acids found in proteins, only cysteine contains a thiol group, and as such, cysteine residues are uniquely capable of being joined together to one another via disulfide bridges. And this molecule that you see down here with the disulfide bridge, this actually has a name. This is cysteine. Okay, so we take cysteine residues and we join them together and we make this cysteine. But nevertheless, disulfide bridges are commonly observed in proteins. And so you can see here that disulfide bridges, like I said, are commonly observed between cysteine residues in the same strand sometimes, or between cysteine residues in different strands. So you see here in these two molecules, in the first one, or I should call them proteins or peptides, okay, but in the first one, we have an intrastrand disulfide bond, right, between two cysteine residues. And here we have it between two different chains, okay, and that is called an interstrand disulfide bridge here. Peptides play a variety of important biological roles. For example, if we take a look at these two pentapeptides that are shown here, we've got leucine, enkephalin, and then we've got methionine, enkephalin here. You'll notice that they both share the same first four residues, right? They've got tryptophan, glycine, phenylalanine. The only difference between the two of them is the C-terminus is leucine in the leucine enkephalin and methionine in the methionine enkephalin. But anyhow, both of these are found in the brain where they function in the control of pain, and it's believed that they interact with the same receptor as morphine. Bradykinin, shown down here on the bottom, is another biologically important peptide. It functions as a hormone that dilates blood vessels and acts as an anti-inflammatory agent. Bradykinin is a nonapeptide, so you can see it's got 9 residues with the sequence that's shown here. So you've got arginine, and then proline, glycine, phenylalanine, serine, proline, phenylalanine, arginine. Vasopressin and oxytocin are very similar in structure and represent 2 more biologically important peptides. Each of these peptides contains a disulfide bridge. Also notice that the C-terminal end in each case has been modified. Instead of having a carboxyl group, you see there's an NH2 group sticking out here and here. And so what that means is that instead of it just being a carboxyl group like this, it's actually a primary amide on the end like there, just a modification. Now, while these two peptides, vasopressin and oxytocin, are similar in structure, only differing in the identity of 2 residues, the ones you see highlighted in red, they nevertheless have very different functions. Vasopressin is released from the pituitary gland and causes the reabsorption of water by the kidneys and controls blood pressure. Oxytocin induces labor and stimulates milk production in nursing mothers. These examples illustrate how a small difference in structure can produce a large difference in function. All right, draw the structure of the following peptide. So remember what I told you, that the sequence always goes N-C-carbonyl, and then you just repeat. So this would represent one amino acid unit. So if leucine, alanine, glycine, and we also want to put the R groups in with the correct stereochemistry. So there's a skill builder that covers this in the textbook, but I'll just go over it with you in this video. So the way it's going to work is like this. We're going to go N, going up, C, carbonyl. Then we're going to go N, C, and this one's going to be going down, and then to our next carbonyl, and then N, and then C, and then carbonyl. Okay? So there's our 3 amino acid residues, or at least their skeletons. So now let's finish up the N terminus, and we'll finish up the C terminus like that. Also, the 2 nitrogens that are in the peptide bonds need hydrogens, so I'll put those there since neither of them is proline. Proline would have, the R group would be tied or tethered into the nitrogen. But anyhow, so we've got leucine, alanine, and glycine. So here's how it works. Leucine has an isobutyl group as its R group. And so if if the bond is pointing up, you make it a wedge. And if it's pointing down, you make it a dash like that. So we're going to put our isobutyl up here for our leucine. Then for our alanine, it's just a methyl group, so we'll just leave it like that. And then glycine, remember glycine is achiral, right? It's just 2 hydrogen atoms that are here, so we leave those out. Now conversely, if you're saying, well, you drew the first bond pointing up like that, Mr. Dion, what if I went down instead? So what if you went NC carbonyl, NC carbonyl, NC carbonyl. Okay, there's nothing wrong with that. You would do the exact same thing. Fill out the nitrogens like this. So now your R group, if it's pointing down, again, it would be on a dash. So we would put our isobutyl group for our leucine, and now our methyl group for our alanine would be in a wedge like that. So both of these, what I've drawn here, both of these are the exact same molecule. Okay, there's no difference between them whatsoever. And the next question is just using 1-letter and 3-letter abbreviations. So again, you don't have to memorize the amino acids in this class. I don't think it's a bad idea because you will have to memorize them for biochemistry. But all we have to do is look at the R groups here. So we can see we have an isobutyl group here. So that represents a leucine. Then we have a methyl group here. So that's an alanine. Then we have a benzyl group here. So that's a phenylalanine, PHE. Then we have a cysteine, so that's CYS. And then we have an aspartic acid up here. So this is the R group for aspartic acid. Now I have a hard time remembering the 1-letter abbreviations sometimes, so I had to look these up. But so we could write down the structure of the peptide like this, like leucine, alanine, phenylalanine, cysteine, and aspartic acid. There's nothing wrong with that, okay? But you could also just write it like this, leucine, alanine. It's F for phenylalanine, then C for cysteine, and aspartic acid is D, okay? So those are the 1-letter abbreviations. But again, you don't have to memorize these. You're always allowed to bring in a handout that I provide into the quiz. So you no need to memorize these, but it's not a bad idea.